everyone, Dr. D here, and in this video I am bringing you the long anticipated Biology 2420 Practical Review. Alright, so we are going to cover material that you should know for the practical. Don't forget that the practical is a written test, meaning that most of the answers are fill in the blank, or maybe even very short answer. There are blanks to fill in, so you do need to know how to spell things. So spelling counts, recalling terms counts. Although there are some multiple choice on the practical, those are the exception. Mostly it's you have to recall terms, fill in terms, remember how to spell terms. So don't forget, study all of that stuff. All right, so for practical 1, it looks like we had introduction, safety check-in, and ubiquity. From those, I would say, and we had mock academic as well, but usually we're not really focused too much on mock academic. It was a nice chance to meet each other. Let's go to ubiquity of bacteria. All right, so from ubiquity, recall that we did learn how to store specimens. Remember, we used agar plates. You should know what an agar plate is. And let me show you. One thing you definitely want to know from this activity, the ubiquity of bacteria, is that there are 2 reasons that we incubate our plates upside down. Do you guys recall what those 2 reasons are? One, remember, one reason was to prevent that precipitation, and another reason was to prevent contamination. So condensation and contamination were the 2 reasons. Remember that? All right. What else? Microscopy use. Microscopy use. Let's look at that. Microscopy use. From this one, you may want to know the difference between the condenser settings, bright field, phase contrast, and dark field. Remember, when you're looking in the microscope at bright field, you know, it's the normal way you see things. Everything is bright. With phase contrast, you have the pH setting on the condenser, and remember, in that case, the specimen is a little bit darker than the background. And in dark field, remember, the background appears completely dark because only light that hits the specimen is reflected into the objective lens. So remember that in dark field, the background's going to be dark and your specimen's going to be bright. Other than that, you should know a lot of the stuff that we covered. How do you calculate total magnification? Remember, total magnification of the microscope is the magnification of the objective times the magnification of the ocular. And remember that ocular is at 10x magnification all of the time. You should know all the parts of the microscope. So see here, right here, this whole thing is called the condenser. But if it's on bright field, there's this little bar here, and that bar, remember, you slide it left, you slide it right. That's the iris diaphragm, and that's for adjusting the amount of light that goes through the condenser. So you should know what every piece is on the microscope and the function. So the condenser, for example, is this whole thing. Its job is to focus a cone of light onto the specimen. The iris is this little bar right here. Its job is to increase or decrease the light, which increases and decreases contrast. When you decrease the light, you increase contrast, right? So it helps with contrast. You should know what the stage is. You know, the stage is the platform on which the specimen goes. The objective lenses, right? You've got the 10x, the 40x, the 100x. The ocular lens is for looking through and for magnifying the image. The objective lens is for collecting light and magnifying the image. You've got the bulb. Remember, you've got the arm here. Number 3 is the arm. Number 12, these are adjusters for the stage. They move the stage around. Number 5 is called the base. This big knob here, remember the big knob is called the coarse adjustment knob. The smaller knob here, this is called the fine adjustment knob. Remember, you only use the coarse adjustment knob with the 4 or 10x objectives. This is the fine adjustment knob. This is only to be used with the this is to be used with all of the objectives, but mainly the higher magnification objectives. What else? Do you guys remember the term parfocal? Parfocal means that the object you're looking at, the specimen, remains nearly in focus. As long as it's in focus with one objective, it'll remain nearly in focus with the other objectives. Yeah, this lever here, or this knob right here, this knob lowers and raises the stage. What else? So again, you should know what all of these bits are. Coarse adjustment knob, fine, the arm, power switch. We went over all of these. Which one's the oil immersion lens? The only one you can use oil with. Remember, that is the 100x. Okay. What else is there? I'm just looking through. You guys know what parfocal is. We just went through that. Why reduce your light with the 10x objective? Because the 10x objective collects more light, so it might be too bright. Okay, so that was microscopy use. I don't know of anything else that you'd really want to focus on. I mean, of course, go through this. Just a word of warning, I may not remember everything that you need to know. I'm just going through some of the stuff that stands out to me, but what I would do is, if I was you, I would study all this stuff, just go through the whole thing. But I'm just touching on some of the stuff that I really expected you to learn from our class. Now, what else? Preparation of specimens. Where is that? Preparation of specimens. Let's click on that. Okay, from this you should definitely know cocci are spherical cells, bacilli are rod-shaped cells. You should know spirillus are spiral-shaped cells. You should know that cocci, bacilli, spirilla, these are shapes, right? So that's shape. But that doesn't tell you about arrangement. Arrangement means, for example, what pattern do these shapes take up? Do they take up chains, or do they show clusters, right? There's terms for that. Remember, strepto means chains of. Staphlo means clusters of. Don't forget, if I'm asking you about shape and arrangement, if I'm talking about this right here, the shape would be coccus and the arrangement would be strepto. If I was talking about this right here, it would be shape, coccus, arrangement, staphlo. Right here, shape, bacillus, arrangement, strepto. Okay, so do recall those terms. What else? Why do we heat fix? Do you guys remember preparing a specimen for smear? You allow the smear to dry and then you heat fix. Do you guys recall the purpose of a heat fix? Well, that was so that we could adhere the bacteria to the slide and also kill them, but mainly to adhere the bacteria to the slide. That way, during the staining process, those bacteria do not wash off. What else? What is a simple stain? Do you guys remember a simple stain? Usually simple stains are done with basic dyes. You guys should remember that basic means positively charged dye. Basic, positively charged. And why do we use positively charged basic stains? Well, because the cells tend to have a negative charge at the surface, right? So the basic dye, positively charged, it gets attracted to cells which have a negative charge due to that membrane potential across the membrane. Okay, that's good to understand. That's good to understand. And again, what is a simple stain? You're staining all of the cells. You're staining all of the specimens. You're not trying to distinguish between this type of cell and that type of cell. Now, the only thing you can tell with a simple stain is shape and arrangement of the cells. Do you think all basic dyes would have the same binding affinity? We don't really get into that. Draw bacillus-shaped bacterium. What would happen if you forgot to heat fix the smear? Remember, it would just wash right off, right? Because it would not be adhered to the cell. Okay, so I think that's about it from here. What do you guys think? Again, please, please, please read through everything, study everything, because there are things I might totally forget to mention. There are things that might be on the practical that I don't even mention in this review. So please study everything, but I'm trying to touch on the more important things that I can remember anyway. Capsule stain. Let's go to capsule stain. Where are you, capsule stain? Here, capsule stain. Do you guys remember that some bacterium are capsulated and some bacterium are not? The capsule stain is a type of differential stain because it allows you to differentiate between capsulated on the left and non-capsulated on the right bacterium. Also, remember that bacteria who have capsules, the capsule itself is non-ionic, which means that the capsule doesn't have a charge. So when you stain with acidic or basic dyes, these dyes stain the cells, they stain the glass slide, but they cannot stain the capsule itself. So here you can see, let me show you, look at this. You can see that the bacteria is stained and the glass is stained, but you see this halo, this clear area around the bacteria? Well, that is the capsule. And here, look here, on a non-capsulated bacteria, you see the background stained, the bacteria are stained, but there's no halo, there's no clearing around each bacterium. Okay, so that's good to understand. What else? What does a negative stain mean? What's a negative stain? Remember, negative stain means you're staining everything but the sample or the structure that you want to see. Okay, see here again, we stained the background, we stained the bacterium, but we did not stain the actual capsule. Motility tests. Let's go over to motility tests. Here. You should know the types of flagellation. Atrichous means no flagella. Monotrichous means one flagella. Peritrichous means flagella all over the place throughout the perimeter. Amphitrichous means a flagella at each end. Lobotrichous means a tuft of flagella, a tuft. Okay, so do know the types of flagellation. You should know what this is. This term here, Brownian motion. So Brownian motion is a fake kind of motility. It's a false motility. Why? Because Brownian motion, what you're dealing with here is vibrations, right? There's water molecules bouncing onto the specimen, causing the specimen to bounce around. And that looks like the bacteria is moving around, but it's actually not. It's just a type of vibration. Remember here, we did two types of motility tests. We did a TTC motility test with the dye or salt, tetrazolium, and we did a hanging drop test as well. So let's talk about the the TTC motility test. You guys remember that was a deep that you stabbed with a perfectly straight stab into the deep agar. The agar was clear at beginning, and then after incubation, wherever the bacterium would grow, the bacterium would reduce their environment, which would cause tetrazoleum to change color, right? Tetrazoleum in this case was a color indicator. It turns red when reduced, and that is as a result of bacterial metabolism, right? So remember, you should remember that this is a negative result for TTC motility because the bacterium only grow along the stab line. This was a positive result for motility because the bacterium were able to grow into the agar, and the only way to accomplish that would be to swim into the agar from this the needle prick, right? So that's a good example of motility on the right. That's an example of no motility on the left. Again, you should know the term tetrazoleum. You should know that it's a color indicator. Okay. What else? We did a hanging drop as well. Do you guys remember? You should know that we used this type of slide. Remember this special slide we used with the depression in the middle? That's called the depression slide. We used a depression slide for the hanging drop test. And the hanging drop test allowed you to see motility. And true motility was seen with cells that run in straight lines and then tumble, right? The flagellar motion involves runs and tumbles. Okay, that's true motility. Remember, Brownian motion is not true motility. All right. So, I think we touched on everything here. What do you guys think? So again, please read through this. Don't take my word for it that we touched on everything, but that's about all I could recall. Counting bacteria. The next one is counting bacteria. Let's find that one. Counting bacteria. Oh, I just noticed I'm going down this side, but there's a whole nother side we need to go down. Don't worry, we'll touch on all the modules. Don't worry, we'll get to everything. Counting bacteria. Do you guys remember the countable plate? It has 30 to 300 colonies on it. So don't forget, countable plates have 30 to 300 on the plate, okay? What else? Oh, do you remember that there were two types of motility? We did a standard plate count, right? The plate count method. And we did the turbidimetry method, turbidimetric test as well. So do you guys remember? I might ask you, or you might need to know, what's the difference between the plate count method and the turbidimetry method in terms of numbers? Why do you always get more numbers? Why do you get more numbers with a turbidimetric assay than you do with a standard plate assay? Do you guys remember that? Well, one thing is, with this one, you're counting cloudiness of the broth, right? So that would include living cells and dead cells. You're always going to get a bigger number if you're counting living and dead cells, assuming that there are some dead cells in there, than a plate count. A plate count, you're not going to see colonies from dead cells. Dead cells don't grow. So you're always going to get a larger count with the turbidimetric test than with the standard plate count test. So you guys should remember how to use the standard formula. Do you guys remember this right here? This is the standard formula right here. The standard formula. You should recall the standard formula, right? You should remember the standard formula because do you guys recall that at the end of this assay, you have to find the plate with the countable number of colonies. The countable number of colonies is the plate that appears to have between 30 and 300 colonies on it. You then have to count the colonies. Do you guys remember the device we used to count the colonies? I don't know if it's here in this packet, but I'll look for it. I don't see it. The device we used to count the colonies, if you recall, was called a Quebec counter. Quebec counter. We used a Quebec counter to count the colonies, and then we plugged the numbers into this equation. Do you guys remember there was a number of colonies, over the dilution factor used. So for example, this bottle would be dilution factor 10 to the minus 6 power times the amount plated. So if we're dealing with this plate, the amount plated would be 0.1 ml or 1 over 10 ml, right? So then you would solve this. This would be, let's say there were 75 colonies. The number of colonies would be 75. The dilution factor would be 10 to the negative 6, or 1 over 10 to the 6, and the amount plated would be 0.1 or 1 over 10, right? To solve that, you would do 1 over 6, 1 over 10 to the 6 times 1 over 10. That's 1 over 10 to the 7. You would then bring that up to the top. It would be 75 times 1 over 10 to the 7, or sorry, 75 times 10 to the 7, 75 times 10 to the 7. Move the decimal point over for standard scientific notation, and that would be 7.5 times 10 to the 8, right? So there you go. That would be your CFUs, your colony forming units. And you didn't actually need to use a calculator. You could just, remember the trick we learned in class, you just add exponents when you multiply these factors, right? When you multiply fractions, you just add the exponents and then you bring it up. So anyway, very straightforward. Again, this counts CFUs. Don't forget the units or CFUs. Don't forget this equation here for the standard plate count equation. Okay, now let's go on to the turbidimetric assay. Do you guys remember what we used? We used a spectrophotometer. Remember this guy? This device here is a spectrophotometer. It measures what? You guys should remember what you're measuring. You're actually measuring absorbance, right? The device measures absorbance. And from absorbance, we can determine concentration, right? So we can determine concentration from absorbance using a conversion factor. Remember, the conversion factor was 8 times 10 to the 8 cells per ml. Or that was your OD600. Your OD600 was 8 times 10 to the 8. You don't need to remember that particular conversion factor. Okay. Now, if you get a graph like this on the, if you see a graph like this is a very straightforward standard curve, right? For example, if your absorbance is 0.05, then how many cells would you have? You could go up from here and across. It looks like you'd have about 12 or 11 times 10 to the 6 number of cells. If you had an absorbance of 0.1, you would go up and over, and it looks like you would have about 24 times 10 to the 6 number of cells. So this is a handy way of looking at absorbance and cell numbers and relating the two using this standard curve. Okay. Again, don't forget you're counting living and dead cells here. And it looks that's about all from there. That was a fun little lab. Next one, effects of temperature. Effects of temperature. Okay, do you all remember? Let's find that. Effects of temperature. Come on, let's see. Do you all remember that bacteria have specific temperatures that they prefer to grow? Some bacteria prefer to grow in cold conditions. These are called psychrophiles. Some bacterium prefer more moderate temperatures, like the human body temperature, mesophile, right? And then some bacterium prefer hot conditions, very warm conditions. Those are thermophiles. And then some bacterium take it to the extreme, and they can exist in temperatures above 85 degrees. Those are your hyperthermophiles, right? So you should know these terms. Also, you should realize that most bacterium that infect humans and cause human diseases are mesophiles. Like E. coli would be a mesophile. Okay. What else? You might get plates, right? Be able to interpret plates, obviously. If you see bacteria grow here but not here, be able to interpret your plates, right? Be able to look at plates and determine if it's a thermophile, a mesophile, a psychrophile, right? What else? I think that's about it. Obviously, if you have test tubes, right, you have like 3 test tubes. Let's say we have 3 test tubes, and the 37-degree test tube is the cloudiest one, after incubation. That would mean it's the most turbidimetric one. That would mean they grew the best at 37 degrees C, right, in a TSB broth. All right, next lab. Hand scrub. Just realize that we used the hand scrubs, and obviously, remember, in ubiquity of bacteria, your hands have lots of bacteria. Everything on you has bacterium. So when you use these hand scrubs, you can greatly diminish the number of bacterium on you for any kind of work you're going to do in a clinical setting. Okay, so that's it for this whole column here on the right. We covered every single lab. Now let's look over here. Transfer of bacterium. Let's look at transfer, you guys. Okay, so again, don't forget, aseptic technique is very important. Why do you work around a flame? Because it's an area where it's relatively sterile. Why did you put your plates upside down? To prevent the condensation and contamination. Do you guys remember that? See, this right here. Contamination and condensation are reduced. What else, you guys? Oh, do you guys remember a spread plate? Kind of a spread plate. The bacterium are on top. A streak plate is different. Where's the... I'll show you a streak plate next time. But you should know for the practical, before I forget, the difference between a streak plate, remember the 3-zone streak plate for isolated colonies, spread plate, which means you're spreading the bacteria all over the agar surface, and a pour plate. Remember, a pour plate, you pour the agar with the bacterium inside with those liquid pores, and then it solidified in the plate, and then you could see bacteria inside of the agar, these little colonies in the agar. So you should know the difference between those 3 types of plates. Okay, what else? Obviously know the difference between a slant and a broth. Know the difference between a slant and a broth. Very good. I think those are the main things from that lab. What else? Pure culture. Let's go to pure culture. This is the one where, yes, the streak plate. Obviously, you should know what a good streak plate looks like. You see, a good streak plate, you have well-isolated colonies in the 3rd zone. You're not going to see colonies in the 1st zone. There's just too many bacterium. You're not usually going to see colonies in the 2nd zone. There's too many bacterium. But you will see colonies in the 3rd zone. Why? Because they become diluted, right? Obviously, what if you got a plate and there were like a bunch of colonies, but half looked creamy and round, and half looked, I don't know, like more yellow and lumpy? Well, that would suggest that there are 2 different species of bacteria on that plate. So just be aware of that. Remember, we did talk about colony morphology. You see here on this streak plate, this is a nice streak plate because I can see all these isolated colonies here. Right, and again, like I said, no pour, streak, spread, and what's the difference between all of these? Okay, very good, very good. Let's go to something else. Gram staining. Let's go to gram staining. Gram staining. You should know all of the reagents of the Gram stain, right? You should know crystal violet is the primary stain. You should know that Gram's iodine is the mordant. Do you guys remember what a mordant is? We talked about this. A mordant keeps a dye in place. You should know that acetone alcohol is the decolorizer here. And you should know that the counterstain or secondary stain is safranin. You should also know that the bacterium are purple if Gram positive and pink if Gram negative. What else? I think those are the big things, right? Those are the big things. All right, next thing. Spore stain. Spore stain. Endospore stain. Here we go. Spore stain. What do you need to know here? Obviously, know all the steps. You know, the dyes, I mean. The dyes. The primary dye was malachite green. Why did we have to heat? Do you guys remember why we had to heat the bacterium with the primary dye? It was to open up the pores inside of the endospores. When the endospores are heated, that expands the endospore pores, allowing the malachite green to enter the spore. Once inside, it's kind of trapped, especially when the bacterium cools down. Now the malachite green is trapped inside of the cell, and when you add water, see, there is no decolorizer. You guys should realize there is no proper decolorizer in the endospore stain. Instead, you simply have water. Water will wash the malachite green out of any vegetative cells. Remember, the vegetative cell is the mother cell, and then the endospores are inside, right? The water will wash the malachite green out of the vegetative cell, leaving malachite green inside of the endospore. Okay, so look at here. You can clearly see that the vegetative cell is stained pink or red with safranin, and the endospores look like little green eggs inside. That is the protective spore inside the endospore. Okay. What's the secondary stain? Safranin, right? So malachite green, primary stain or primary dye. Decolorizer, there is no decolorizer. It is simply water. And then safranin is the secondary stain. Okay. Okay. That is about it for that one. Let's go to acid-fast stain. Acid-fast stain. What's the primary dye? Carbofuscin. What's the decolorizer? Acid alcohol. Acid alcohol. What's the counter stain? What's the counter stain? Oh, it's methylene blue, but where is it written here? The counter stain is methylene blue. Okay, so you should know the primary stain, the decolorizer, the secondary stain or counter stain. Remember, secondary stain and counter stain mean the same thing. You should also realize that the fuchsia colored or purple colored bacterium are acid fast. The light blue colored ones are non-acid fast. You should know why bacteria are acid fast. Do you guys remember why bacteria are acid fast? Because their cell walls contain this mycolic acid, which is a lipid in their cell wall. And this lipid is very waxy, and it is what gives these bacterium their property, their waxy property, and their ability to be acid fast, right? So, good things to know. Good things to know. What else? Dilutions and pipetting. Let's get to that. All right, dilutions and pipetting. You should know how to read a pipette from the bottom of the meniscus. You should know how volumes work in a pipette. So, for example, it's two different things. Look at this pipette. How does the volume work? There's two different questions I could ask. I could say, how much is in this pipette? How would you answer that? Well, how much is in this pipette? If this is a 10 ml pipette, the amount that's in this pipette, people look at this and they think, oh, it's, I don't know, 9.9. That's completely wrong. Remember, the scale is inverted on these pipettes. So the amount that's in this pipette would be 1, almost 2. It's 1.9. Because you read from the bottom of that meniscus, right? So, but that's a different question than asking how much has been dispensed from the 10 ml pipette. If that's the question, that means we started at 0 ml and we dispensed down to this point. So how much would have been dispensed? In that case, it would be 8.1 was dispensed. Okay, so very good to understand, right? Okay, what else do we need to know? Dilution factors. Do you guys remember how to do a dilution, right? If you add, let's say if you add 0.5 ml of this blue dye to this tube, and the tube has 4.5 ml of water, what kind of dilution is that? You could call it a 0.5 in 5, right? 0.5 divided by 5. Or you could simplify that and say it's 1 in 10, right? Because 0.5 over 5 is the same as saying 1 in 10 dilution. That's called getting the dilution factor. Now, how do you get total dilution factor of a serial dilution? You see, let's say we take some of that 1 in 10 dilution and we add it to the next tube. Well, let's figure out the dilution factor of the next tube. Remember, 1 ml inside of 9 means 1 in 10. So this is a 1 in 10 dilution. This is a 1 in 10 dilution. How do we get total dilution factor? You multiply them. So 1 in 10 times 1 in 10 equals 1 in 100, right? So you should know how to get dilution factor, and you should know how to get total dilution factor of a serial dilution. Okay, also we talked about this before, but the standard plate count, the number of colonies on a countable plate divided by total dilution factor times amount plated, right? So you should be able to, for example, solve this problem here, right? You should be able to solve this problem here. Okay, and express your value in terms of scientific notation, in the form of scientific notation. Very good. That's about it from this activity. Okay, what else? Colony morphology. Colony morphology. Let's look here at colony morphology. Now again, remember that bacteria tend to have a particular colony morphology. Look at this. If you got this plate, for example, if you got this plate and the question was how many different species appear on this plate, what would the answer be? Well, to answer that, you would have to say, well, how many different looking bacterium are there? So notice how all of the threes, you see all of the ones called 3, notice how all those colonies have the same morphology, the same colony morphology. So they're probably all the same species. Notice the 4s. You see how the 4s have the same morphology? That would mean that they have all the same. They are probably all the same species. So you could basically say that on this plate there are 8 different species. That obviously there's more than 8 total colonies, but if you're talking about types of species, you should realize that each species of bacterium has one particular colony morphology, like how it appears on a plate when it forms a colony. And remember, colonies are clonal, meaning that they came from one original bacterium that divided through binary fission to give you a colony of bacterium. Okay, now these are the properties here of colony morphology: chromogenesis, opacity, elevation, surface. Now you don't need to go through and memorize all these types of things like ground versus regular versus filamentous. Like, that's a lot of work, you know. Yeah, I don't want to overwhelm you. Just realize that different bacterium have different species colony morphologies, And those can involve these traits here. That's all. Now, environmental conditions and growth. Let's get to that. Environmental conditions and growth. Do you guys remember what we did here? We tried bacterium osmotic pressure tests. We grew bacterium on plates of varying concentrations of salt, 0.5% salt, 5% salt, and really high 10% salt plates. Bacterium that can grow in high salt concentrations are called halophilic bacteria or haloduric bacteria if they can tolerate high salt. Remember, that's why Staphylococcus aureus or Staphylococcus epidermidis, these are bacterium that grow on the human skin. And that's because they like that salty skin. They can grow in the salty skin, whereas bacterium that cannot withstand high osmotic conditions, high salt conditions, the ones that are not halophilic can't, right? So just remember, you might get plates to look at, and plates, high salt plates, low salt plates, and the questions, you should be able to address, which of the bacterium are halophiles, which one is not, right? Which one's growing in the high salt plate, which one's not growing in the high salt plate. You should know what that term halophilic means, okay? Remember, we did a UV experiment as well with different plates. Do you guys remember 10 seconds, 60 seconds, 5 minute, 10 minute? The more time in UV, in direct UV, the more death occurred and the less growth occurred. But do you remember in the 60-second plate with the lid on, the bacterium grew just fine because the bacterium were untouched by UV. The plastic lid blocks the UV. And on the 10-minute plate, you guys remember we put a index card on one half of the plate and not on the other half of the plate? You should know on which half of the plate did the bacterium grow and why did that happen. So remember that. Do you guys remember we did pH tests? We had 3 different broths, a pH 3 acidic broth, a pH 7 neutral broth, and a pH 10 basic or alkaline broth. You guys should realize that basic means alkaline, okay? So bacterium that grew best and produced the most turbidity at pH 3 are called acidophiles. Bacteria that grew best and showed most turbidity at pH 7 are neutrophiles. Bacteria that grew best at pH 10, those are called the alkalinophiles because they prefer basic or alkaline conditions. Okay, and that was about it from that activity, right? Very, very good. Let's go on to Kirby-Bauer test. Okay, Kirby-Bauer. Talk about the Kirby-Bauer test. Antibiotic sensitivity test is called Kirby-Bauer. This is a disc diffusion test with different, do you guys remember, we used different discs. Each disc had a different type of antibiotic on it. So what we did was we spread bacteria on a spread plate. We spread bacteria on a TSA plate, and then we placed various antibiotic discs on top. And the antibiotic would diffuse into the agar surrounding the disc. And the bigger the zone of inhibition, remember zone of inhibition is this zone around the disc. And to measure the zone of inhibition, remember you measured the diameter in millimeters from one end of the circle to the other end of that circle. That zone of inhibition, the bigger that zone is, the more susceptible the bacterium are. You see, look at this. Look at disc C. The bacterium are growing right up to disc C. So the zone of inhibition would be 0 and the bacterium would be considered resistant. Look at disc A. The zone of inhibition is quite large. If you were to measure it with a ruler, you would measure from where my pointer is here to where my pointer is here, and you'd get that diameter in millimeters. And that looks like the biggest zone of inhibition. So these bacterium are quite susceptible to disc A, right, to antibiotic A. Do you guys remember how to use, here's an example of using a ruler. You would measure from one side of the diameter to the other side. So I would say it's probably starting right here at the 3, and it's a couple notches after the 4. So that would be 10, 11, 12, 12 millimeters across. This would be 12 millimeters across. And this is what, GM, gentamicin, right? So GM, 12 millimeters. Let's go to our table here. Gentamicin, 12 millimeters means resistant, okay? So this is actually, this bacterium, even though there's a big zone, it's not susceptible. It's still considered resistant for gentamicin. See, gentamicin 12 is resistant. So you get the diameter in millimeters, you compare it to the table here for that particular disc. See, GM means gentamicin, 12 millimeters means resistant. So these bacterium are able to resist that antibiotic, right? So very good. So you should know what a zone of inhibition is. You should know how to measure the zone of inhibition, and you should know how to look up whether the bacterium is resistant, intermediate, or susceptible. And you should know, obviously, the name of the test, the Kirby-Bauer test. You should know what that is. Okay, next thing. Antimicrobial chemicals. Antimicrobial chemicals. What did we do here? We tested various chemicals. We looked at the zone of inhibition there to see how these chemicals affected bacterial growth. Okay, I don't think there was anything very special here that wasn't in the Kirby-Bauer. Again, know what a zone of inhibition is, and I think you'll be good. Did we miss anything? Oh, protozoa and fungi. Let's do protozoa first. Protozoa. All right, protozoa. You should know that protozoa are categorized by what? Do you guys remember? Protozoa are categorized by their motility. You know, yes, cell structure, but also motility, right? Some bacterium move around with pseudopods. Some are flagellated. Did I say bacteria? I meant protozoa. Some protozoa, like the amoeba, they have got the pseudopods. Some protozoa are flagellated. Some are ciliated. Some can't move at all, actually. They're intracellular parasites like Plasmodium. Okay, so protozoa are categorized by their mechanism of motility. You should know what amoeba look like, what paramecium looks like. You should know trypanosoma looks like this, is flagellated. The trypanosoma, by the way, you see all this in the background? These are all red blood cells. Trypanosoma are these whip-like creatures, these whip-like protozoa, and they live amongst your blood cells, but not inside of your blood cells. You see that? They can be pathogens, animal pathogens. They live in your blood, but not in your blood. Look at this. Plasmodium, on the other hand, over here, Plasmodium lives inside of your blood cells. Now you've got your blood cells, and look, these little ring-shaped structures inside. It looks kind of like eyebrow rings or belly button rings. You know what I'm talking about? These, or headphones. You know, these look like little headphones. These little guys are the Plasmodium. And notice how the Plasmodium lives inside of the red blood cell, whereas the trypanosoma lives outside of your red blood cells. They're both blood pathogens, but one is an intracellular pathogen, Plasmodium. One is a extracellular pathogen, trypanosoma. Okay, what else? So amoeba, paramecium, Plasmodium, trypanosoma. Those are the things to know. Those are the things to know about. All right, so yeah, know the structure under a microscope, what trypanosomes look like, what Plasmodium looks like, paramecium, amoeba. Just know what they look like under a microscope. Very good. Next thing, fungi. Let's talk about fungi real quick. Fungi. So fungi are not photosynthetic. Fungi are saprophytes, which means they break down dead material, dead organic material. You should know about Saccharomyces cerevisiae, which is baker's yeast. You see, yeast grow as these oblong-shaped cells. You see, they're kind of round-ish, like almost egg-shaped, these oblong structures. These are yeast, right? You should know about penicillium. Remember penicillium? You've got these finger-like projections. And then aspergillus, on the other hand, the spores form what appear to be like fans. So here's the difference between penicillium and aspergillus. You should know the structural differences of the spores of penicillium and aspergillus. So look, penicillium, the spores are growing out like finger-like projections like this, right? Whereas in aspergillus, the spores fan out more like a peacock's tail, like this. Okay, so that's good to know. Let's look at Rhizopus solanifer. Rhizopus is bread mold. You might want to know about the bread mold. Do you guys remember Rhizopus can form sexual spores where hyphae from different mating types touch and they form a sexual spore called the zygospore. And that's what a zygospore looks like. That's what a zygospore looks like. These are sexual spores. And they can also form asexual spores. These look like little microphones. Look like a little microphone. That's not a zygospore. That's not a sexual spore. That's an asexual spore called sporangiospore or sporangia, right? So you should know that this is the sporangia. This is the zygospore of the Rhizopus solanifer, which means the common bread mold, okay? What else do we have? We can have, these are the big ones, actually. Trying to see if there's anything really else that's very important. No, I think we've touched on most of the important things. And again, this leads me to the end of this practical review. But please, please, please realize that I am but a humble human and I can make mistakes, okay? And I may or may not have remembered everything to touch on for the practical, but these are the big things that, if you recall from lab, we stressed and we focused on and we saw. So please, please, please study everything. Don't, just go off of this review. I tried to remember a lot of the important factors for you to remember for the practical exam, but it's really, really up to you to go through your packet here of notes, go through all of these different modules, these lab modules. Make sure that you really, really, really study them and you know everything and you know how to spell things and you know how to answer all the questions and remember what tetrazolium dye is and you remember all those terms that are really easy to forget and difficult to remember. But that comes with a lot of study, time, and effort. So thank you for watching. I hope you do really well on the practical. Let me know if you have any questions, and I'll catch you next time.